Okay, this is question 6 on the A4 mock for poorly people. So, what is a reflex action? This is straightforward. AO1. Have you learnt the definition out of your notes? If you have, brilliant. 2 marks. So, rapid response to a stimulus. Involuntary. That means you don't have to think about it. So that's great. Again, you know, AO1 all the way. So this is a beautiful, easy question. Name the types of neurone. Now in this case you do have to do a bit of looking at the diagram. So here we've got the sensory receptor. So this is kind of upside down from how it was. So here you're going to have your effector at the other end, which means that A is your motor neuron. C is going to be your sensory neuron, and the other sort of dead giveaway on that one is that it's going through this structure here, the dorsal root ganglion. And then in between we've got the relay neuron. No real excuses for getting any of that wrong. And the same here, AO1 again. Can you label? Well, you should be able to. So these are the gaps where the axon's exposed. Node of Ranvier. Ah, beautiful word. Ah, Monsieur Ranvier! And here you could either have a myelin sheath, or you could have had the cell that produces it. You are expected to be able to spell Schwann. Another great word. So, so far all AO1. A little bit trickier now describing how a resting potential, now this is in your notes again, I would say this is AO1, but potentially AO2 because you have to do a bit of question interpretation. So resting potential, maintained, what do we know about resting? It's minus 70 millivolts, and how do we bring that about? So this is where we need to talk about sodium potassium pumps. Don't forget to give them your charges. So the sodium potassium pump moves 3 sodium ions out of the axon and 2 potassium ions into the axon. And again, this is kind of, you know, have you learnt your stuff? Potassium ions leak out of potassium ion channels faster and sodium ions leak in. So again, without learning the stuff, you've no chance of doing any interpretation whatsoever. Then describe how the potential is reversed. So describe how it goes from minus 70 to plus 40 when an action potential. So they are, it's just saying, yeah, describe the action potential. So a stimulus opens some sodium ion channels. Notice that you are allowed to use the chemical symbol because it's science. If threshold is reached, sodium ion channels open, and sodium ions fusing rapidly and you need to have that word rapidly in there really. It's an influx, which is rapid movement. Okay, now we're going to be a bit trickier. One symptom of Alzheimer's disease, that's a bit of a weird spelling of it, I want to put a T in it, but anyway, is a reduction in the level of an enzyme in the brain that synthesises acetylcholine. So, describe the function of acetylcholine at a synapse. So what do we know about acetylcholine? It's only asking, what does this do? What's its job at a synaptic junction? So, acetylcholine is a neurotransmitter. It is released from vesicles in the synaptic knob, great word, and the vesicles fuse with the pre-synaptic membrane, and I'm just going to put in there, because of calcium ions, the acetylcholine diffuses. So they're really testing here, do you know that sequence, you know, can you use the right words across the synaptic cleft and binds to receptors on post-synaptic membrane. So the terms that you really need to use properly, you need to be talking about the release from vesicles. You could have talked about exocytosis, but you must have these things in order. Presynaptic membrane, synaptic cleft, postsynaptic membrane. Remember what's where. So the sodium ion channels on the postsynaptic membrane open, and depolarization postsynaptic membrane occurs. Okay, turning over, we've got an exciting diagram. Okay, there's that weird spelling of Alzheimer's again. So what have we shown here? The diagram shows neurons from the brain of a healthy 70-year-old. That's A, so this is healthy, and this is B, somebody with Alzheimer's. Would you put a T in Alzheimer's? No. Me neither. Doesn't it just not right, is it? No, looks weird. Anyway, So what do we notice about these? So this is very branchy, isn't it? We've got 1, 2, 3, 4, 5, 6 branches. 1, 2, 3, 4, 5, 6. So just the same. But these branches seem to be much shorter, and they don't have as many sub-branches on them. So we've got less branchy. Don't seem to be reaching as wide an area. They're shorter, and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 synaptic knobs on this side, and 3, 6, 8, 11, 13, 14, 15, 16, 17, 18, 19, 20 on that side, so only half the number of synaptic knobs. So what are they going to ask us? State 2 differences. Well, that's really easy. So they don't want me to give any reasons, they just say, you know, what are they? What are those differences? Can you spot the difference? So there are fewer branches,
Dr. Aloysius Alzheimer, without a T.
Yeah, no T. No T in Alzheimer's.
It's a name, so no interpretation.
No. Fewer branches in the Alzheimer's sufferer. And less synaptic knobs. And the, what should we call them, branches. Oh, I'm going to call them branches again because that's what they've called them. Branches are shorter. Suggest how one of the differences could account for the reduction in acetylcholine. Well, what do we know about acetylcholine? It's made in the synaptic knobs, so less knobs, less acetylcholine, presumably. So as there are less synaptic knobs. I do have a friend who's a biology teacher who can't say the word synaptic knob without laughing. Don't know why. She can't say the P word without laughing either. There are less synaptic knobs.
What about cleft?
Not so bothered. Less acetyl choline is present. Excellent. 13 minutes are good.